The REPLICA project was born from an idea of COIAR, the Center of Excellence for the Acceleration of Harm Reduction of the University of Catania. REPLICA is a 3-year project with excellent partners from different parts of the world. The no-smoke team of the University of Patras from Greece, the Sbarro Health Research Organization of the Temple University from the United States, the Sultan Qaboos University from Oman, the University of Kragujevac from Serbia, the Federal University of Kazan from Russia, and the University of Pajajaran from Indonesia. The first year of REPLICA involved only 5 partners. Italy, obviously, the University of Patras, the Temple University, Sultan Qaboos University, and the University of Kragujevac. Detailed information on the project is available on this link on the project website. All the centers were equipped with the smoke laboratory capable of working in ISO conditions with the same smoking and vaping machines, the same climatic chambers and incubators with exposure systems in order to expose the cells to smoke and vapor at the air-liquid interface. This is an example of the Italian lab in the University of Catania with the smoking and vaping machines on both sides of the controlled temperature incubator and an adjustable aspirator above to eliminate the waste fumes. For the first year of REPLICA, we have chosen to replicate 3 studies on the cytotoxic and inflammatory effect of smoking and vapor on human bronchial epithelial cells, the H292 cells. We have selected the studies because they were conducted by the same research group and have the same setting, but they are conducted with 3 different products. In the first paper, the P1, the cellular model used is studied in depth, and the differences between different regimes of exposure, ISO and ACE, to cigarette smoke are evaluated using the reference cigarette 3R4F of the University of Kentucky and comparing the cytotoxic and pro-inflammatory effects of wall smoke and vapor phase of smoke obtained eliminating TPM and nicotine using Cambridge filter pads. Here we are focusing on the cytotoxic effects of this product. In fact, in the paper 2, the same cytotoxic assessment were conducted using 2 different tobacco eating products. And in the third study, the same assessment are repeated by exposing the cells to the vapor from 2 different electronic cigarettes. We had some differences between the smoking machines and electronic devices used in the original studies and in our study. For example, the smoking machine used for the original studies permit to dilute smoke and vapor with air, while our machine deliver only undiluted smoke and vapor to the cells. Moreover, for some products, we have only found the updated version on the market. As an example, the 1R6F instead of 3R4F, the IGOS 3 instead of the IGOS 2, and so on. Therefore, probably with a different performance in terms of nicotine release. So we firstly had to assess the dose response curve of the cells with our equipment and to define our IC50, the concentration of smoke that is required for 50% inhibition in vitro. Therefore, we measured the nicotine released in the culture media in the cell exposure chambers, determining the nicotine value released by the 1R6F cigarette to reach the IC50 with our cells, and we have exposed with all the non-combustion products to a number of puffs needed to reach the same amount of nicotine in the culture media of the exposure chamber. Oh, sorry. Only wall smoke exposure led to detectable quantity of nicotine in the culture media, confirming that Cambridge filter pad is capable of retaining most of the nicotine without making it arrive in the exposure chamber and therefore on the cells. With the wall smoke exposure, we observed an increase in nicotine concentration with increasing puff number and more with the Health Canada intense regimen than with the ISO regimen, as expected. The TPM release measured by weighting the Cambridge filter pads before and after exposure under Health Canada intense regimen was also significantly increased compared to the ISO regimen. Using nicotine dosimetry data from wall smoke exposures, cell viability of the biological responses was presented against exposed nicotine in the cell media for each puff number. Therefore, the IC50 for nicotine from wall smoke under the ISO regimen was 4.18 micrograms per milliliters and was roughly the amount of nicotine released by 10 puffs. Otherwise, under the Health Canada intense regimen, the IC50 was 9.7 micrograms per milliliters and was reached after about 5 puffs. Based on calculated nicotine IC50 dose by wall smoke under the Health Canada intense regimen, we choose to conduct the experiments with the ends by using the number of puffs able to produce the amount of nicotine closest to this value across all products. Thus, for all the comparison experiment between the cigarette and the ends, the 1R6F was smoked by the LM1 smoking machine with the Health Canada intense regimen for 5 puffs. The vape pen 3 button activated cigarette was vaped by LM4E vaping machine like all the other hands, following a modified Health Canada intense regimen with 1 second of pre-activation for 10 puffs, as per the original paper. The Vape Stick Max, a puff-activated cigarette-like product, was vaped following the Coresta reference method 81 regimen for 25 puffs. The IQOS 3 was vaped using the Health Canada intense regimen without blocking the filter vents for 7 puffs, and the Glow Hyper was vaped by the same conditions for 8 puffs. For each exposure run, we prepared 3 contours: an air contour with the cells exposed to the lab air filtered with a Cambridge filter pad, an incubator, and an air-liquid interface control prepared for each exposure by placing cells respectively with and without the inner culture media in the incubator. Substantial interlaboratory variability between the 5 laboratories was observed for all ISO wall smoke exposure conditions. Linear regression analysis of ISO vapor phase dose response curves showed good reproducibility between each laboratory except for one. Variability in the results for Health Canada intense wall smoke exposure was observed only for 2 and 4 puff in one lab. We evaluated the laboratory performance for each exposure condition and product variability. Interlaboratory variability is low across all laboratories and products except for 1R6F exposure performed by one lab. 1R6F exposure showed the greatest interlaboratory variability. Indeed, interlaboratory variability deviation were greatest for 1R6F compared to other products. There was an overall significant difference in natural red uptake cell viability among all products compared with the air control, driven mainly by the effect of 1R6F cigarettes. No reduction in cell viability was observed after exposure to tobacco-eating products or electronic cigarettes. Thus, in comparison between products, only 1R6F cigarette differed significantly from the other products. Our results support the reduced potential of electronic cigarettes and tobacco-eating products relative to conventional cigarette in an in vitro model of bronchial epithelial cells, even under more extreme exposure conditions, like undiluted aerosol and smoke, than those of replicated papers. For future studies, our findings suggest that aerosol dilution and normalization of data to better reflect cell functionality rather than viability will be important for achieving the most accurate results. Also, an important milestone was the establishment of collaboration within the scientists that will expand teamwork and progress knowledge in the field of reduced risk, non-combustible tobacco and nicotine products. Overall, this study confirmed that most of the harm to bronchial epithelial cells arose from volatile compounds in cigarette smoke rather than TPM or nicotine, and support that use of ENDS in harm reduction strategies for smokers. Thanks for your attention.